Good morning, my dear friends. In the last class, the world of microbe, we have seen that four type of microbes. They are bacteria, fungus, virus, and prions. We had discussed this in the last class itself that these microbes, some of them are beneficial for us. Some of them are very dangerous. So in this aspect, we are going to start this lesson looking at the beneficial aspects of these microbes and how are they useful for us. That is what this lesson we are going to talk about. These microbes are helping us in lot of different things, like some of them are very useful as a biopesticide or as a biofertilizer in the field of agriculture. Some of them are helping us in the industries like preparation of beverages, preparation of medicines, like that. Before we start this session, I would like to tell you a very important concept you should learn. That how you write a scientific name. First, what is called a scientific name? A name which is throughout the globe the same. That actually shows in what generic this organism belongs and what type of species is this. For example, for human being, the scientific name is Homo sapiens, and for a mango tree, it is called Mangifera indica, and for the yeast, it is Saccharomyces cerevisiae. So many names are given to living organisms. Those type of namings are called scientific name. There is an important aspect you should always remember. A scientific name has two parts. The first word is the generic name, second word is the species name. Homo sapiens, Mangifera indica, like that too. And there are certain rules that you should follow when you are writing it or typing it, something like that. That rules you must know. That is a very basics of biology. So that is what we have to discuss, because in a quick way we will finish this. First rule is for writing or how we will print it, these scientific names. I told you there are two names. The first one is generic name and another one is species name, the second one. A generic name has to start with a capital letter and the species name has to start with a small letter while printing. And while printing, we have to follow one more rule, that these two words should be in italics, that means slanting letters. That's all about in printing. First letter of the generic name should be capital and second word should start with a small letter, and both these words should have a space in the middle and they should be in italics. That is a rule you have to follow when you're typing it or you're going to print it or something like that. Okay, what are the rules for writing the scientific names? So I'll be personally looking into your notes how you are going to present it. Whenever you are writing a scientific name, don't forget to write the same rules, like generic name start with a capital letter, species name start with a small letter, but italics is practically not possible. Am I correct? So instead of making it italics, while writing, you should underline both the words separately. So let me conclude how we will write a scientific name. A generic name and a species name should have a space in the middle. And the generic name should start with a capital letter, species name should start with a small letter. And you should underline these two words separately. That is the rule you have to follow always whenever you are writing a scientific name. Okay, let's start this content of this session. how these microbes are useful for us. These microbes are useful for us. What are the uses of these microbes? When we look at that, in the field of agriculture, these microbes are playing a very important role by means of biofertilizers, bioinsecticides, and nitrogen fixation, and some recycling of some elements like that. In three different ways, they are mainly helping us in the agriculture. Let's see them one by one. Like, these microbes are acting as biopesticides or biocontrol agents. What is the meaning of biocontrol? You know that for every farmer, the main threat is from the pest. Some insect will come and it will start feeding on the flower or some the fruit, and the farmer will have huge loss on the profit which they are about to get from the agriculture. So there is a main use of that, use some poisonous chemicals and kill all these pests, the insects which are damaging the paddy field or whatever agriculture activity is there. But remember, using too much of chemicals on these items which we are going to eat it later, which will damage our health. So it is always advisable to go for biocontrol. That means if this is a pest, who is the enemy of that pest? Find it out and use this fellow against that fellow. My enemy's enemy is my friend. That's all the logic. Biocontrolling mechanism, an example is bacillus. A best example for this biopesticide is Bacillus thuringiensis bacteria. This bacillus bacteria producing a protein called cry proteins. This cry proteins, this special proteins produced by this Bacillus thuringiensis, we shortly call it as BT. This bacillus cry proteins are very dangerous for these pests. How? You know that almost all the insects, they will in their life cycle, example a butterfly, they have a larval stage, a worm, a larval stage. So most of all the insects are going through a larval stage. These cry proteins, irrespective of what type of pest it is, this will go and destroy the larva. This larva is about to become a pest which is going to destroy the agriculture. So uses of this Bacillus thuringiensis in the agriculture field, it always kills the pest that actually going to destroy our agriculture field. This is an example for biopest control. The second most important uses of these microbes in the field of agriculture is we can use these microbes as biofertilizers. Okay, come on, what is the meaning of biofertilizers? What is the meaning of fertilizer first? Fertilizers are something, a manure we give to the land or the field so that it improves the quality of the soil in such a way that these plant can grow very well in that particular and give good yield for the farmer. Okay, if you are going for chemical fertilizers for a long time usage, that actually will destroy the land itself. After certain years, you cannot cultivate anything in that because the land is already destroyed by means of increased pH, increased poison, increased unwanted chemicals and all. So there is a better alternative that the nature itself gave is use the bacteria or fungus that are capable of fixing so much of nutrition to the soil, the plant can easily absorb it. So one example is the nitrogen fixation. So before we go to that, you can have a small experiment. If there is a pea plant, pea beans, the plant, if you pluck it, you can see that on the roots, you can see so many ball-shaped bulgings. If you crush it and see it under a microscope, you can see that there are minute bacteria are moving inside. Bacteria are living along with the plant by helping the plant to get the atmospheric nitrogen. What is called nitrogen fixation? You should know that for the good growth of a plant, the plant requires a large quantity of nitrogen. Okay, it is simple. In the atmosphere you have plenty of nitrogen, just take it. No, the plant cannot absorb the gaseous nitrogen as such inside and utilize it. So the plant need the nitrogen in a huge quantity, but it cannot absorb from the source, which is in gaseous format. Someone has to help it by means of converting this atmospheric gaseous nitrogen to a format that this plant can absorb it. There comes the nitrogen fixation. Some bacteria, some are free living, some are in symbiotic association with the plants. They can help to fix this atmospheric nitrogen in such a way that the plant can absorb it by converting this nitrogen into some other nitrogenous compounds. For bacteria like rhizobium, they are living along with the plant as inside the root nodules. They can fix this. And if you look at the free living bacteria like acetobacter, they also perform the same thing, that they also fix atmospheric nitrogen to the soil so that the plant can absorb it. So in such a way that so many microbes are helping in the biofertilization process that actually help the plant to grow very well. The third use of microbe in the field of agriculture is that these microbes, they are helping in the cycling of so many things, because the microbes act as a good decomposers that actually degrade the waste vegetables, the plant which has fallen down, everything will be decomposed, and what are the nutrition stored inside will be dissolved to the soil so that the next plant can absorb it and start growing out of it. And these microbes are also helping in cycling of various elements like oxygen cycle, nitrogen cycle, like that. They all help in the cyclic movement of elements, as well as they help in good decomposition, which is a lot more helpful for absorbing the ready nutrition for the new upcoming plant. To conclude this, these microbes are helping in agriculture by means of three things. First one is biopest control. Second one, biofertilizers. Third one is decomposition and cycling of various elements inside the soil. If we look at the next important part of this lesson is how these microbes are helpful for us. So in that aspect, we are going to look at these microbes, how they are helpful for us in the field of agriculture. That is the first point. For example, they are helping us as a biocontrol agents. You know very well the pest, just insects that will come and destroy the agricultural products. So using chemicals against them to kill it is very dangerous, because in future that will be dangerous for us also, because we are going to eat the products of agriculture. So we can't keep using the chemicals. Instead, we can use some microbes. How it is possible? For an example, Bacillus thuringiensis is a bacteria that is actually producing a protein called cryproteins. These proteins destroys the larva. You know that most of the insect, they will go through a larval stage in their life cycle. So whatever insect, pest it is coming to the agriculture field, they will go through a larval stage. During that larval stage, these cryproteins will go and destroy them, so that you don't need to use any chemicals. Instead, you can use it as a biocontrol agents. These microbes will help us in agriculture by this method. Next one is as biofertilizers. What is the meaning of fertilizers? Something that improves the quality of soil, that you get a better quality of agricultural products. Some microbes like rhizobium, acetobacter and all, for an example we are saying this, there are many. These will fix the atmospheric nitrogen, which is gaseous in stage, which cannot be utilized by this plant. In fact, the plants are in need of huge quantity of nitrogen, but it is available, but it cannot get it. So some microbes are helping it, converting the atmospheric oxygen into a format that these plants can absorb it. For example, rhizobium bacteria will convert it by staying along with the root nodules of pea plants. And some free living bacteria like acetobacter, which is present in the free living format on the soil, can also fix the atmospheric nitrogen, so the soil become fertile. So this is the second use of microbe, that they can be utilized as biofertilizers. The third point is, these microbes are helping in the decomposition of organic material, plant or another microbes or animals. The dead body can be decomposed so that the nutrition stored inside the plant body or the animal body can be dissolved into the soil, so that a new plant can start growing so fast. And these microbes also helping in cycling of the elements like oxygen cycle, nitrogen cycle. You would have studied carbon cycle and all. So all these are done by these microbes, which are very useful for the agriculture field. So let me conclude. Microbes can be used as biocontrol agents. Microbes can be used as biofertilizers. Microbes also helping in good decomposition, improving the quality of soil, also the cycling of various elements like carbon, nitrogen, oxygen, like that. And if you look at the next use, these microbes are helping us a lot in the various industries. What are the industries? For example, we are getting the help of a microbe, Saccharomyces cerevisiae, yeast. Of course, there are two to three type of yeast species, but Saccharomyces cerevisiae, that species helping us a lot to prepare the beverages, preparation of alcohol, wine and all these things. These microbes are helping us a lot. If you look at another use of a microbe in industries is, so the second use of the microbe, and an example is curing of coffee beans, tea leaves, tobacco leaves, like that. You know that coffee beans, tea and tobacco all supposed to undergo a few steps of process so that a customer can use it. You cannot directly use the tea leaves as such. You need to make it some process. That process we use bacteria to give them good smell and good quality also. So curing of these products, we are getting the help from the microbes. Example is bacillus. The next usage of microbe is preparation of curd. Lactobacillus, it actually helps us to convert the milk into curd. That is a use. Curd in industrial production, we have curd packets we are available, and who is helping it? Keeping the milk as such for a long time, it may not give you a good curd, but adding lactobacillus give you good quality of curd. That is a good help from these microbes. And if you look at another example how these microbes are helping us in industrial level is production of various organic acids, vitamins and some enzymes. For example, oxalic acid, all these citric acid, all can be produced with the help of microbes. And the case of protease, lipase, these kind of enzymes can be also produced with the help of microbes. And for a good example, this yeast is very good source of vitamin B complex. On all these ways, these microbes are very much helpful for us by the means of industry, because you are going to get these enzymes or these organic acids or vitamin B tablets. All these are, you're going to get it in industry. Someone has to produce it, so you go and purchase it. So it's an industrial use of all these things. So if you look at the second application of microbe, it's in the field of industries. Examples, I mentioned few examples. Preparation of wine, beverages has helped by Saccharomyces cerevisiae, an yeast. And curing of coffee beans, tea, tobacco and all is with the help of microbes. And so lactobacillus helps us to prepare the curd from the milk. And looking at this next uses, production of various organic acids like oxalic acid, citric acid, all these are produced by various fungus. We can extract it and use it. And looking at something else like enzymes and vitamin B complex, they are produced mostly by yeast and other microbes. So industrially, all these products like alcohol and tea, tobacco and coffee beans and this curd, precious acids, enzymes, vitamins, all these are very precious things we can prepare with the help of microbes. They have industrial money value, you can sell it. So that is how the second application of microbe in industries help us to make money out of it. In the next topic, we are going to discuss that how these microorganisms are useful for us in the field of medicine. In the field of medicine, these microbes are helping us in the preparation of few antibiotics, vaccines and few medicines. And look at the antibiotic first. What are antibiotics? That is the first thing. Antibiotics are products of the microorganisms itself that will prevent the growth of other microorganisms. How about that? It's a wonderful thing, am I correct? That the bacteria itself producing something that will prevent the growth of other microorganisms. And we extract that, we are using it to kill all the microbes if there is an infection inside our body. So that will go and kill other microorganisms like fungus or bacteria. So if you look at an example of this kind of antibiotic by a bacteria is called streptomycin, an antibiotic produced by the bacteria, and penicillin, another antibiotic produced by a fungus. So these are helpful for us in treating any bacterial or fungal infections inside our body. So that is why these microbes are helping in preparation of antibiotics. The second use of this microbe is that this microbe in the field of medicine, we can use them as for the preparation of vaccines. What is called a vaccine? A vaccine is a biologically prepared thing. When it is injected inside our body or through any means, by injection or oral or something like that, if it goes inside our body, it will trigger our immune system in such a way that to fight against in a maximum performance. How do we increase the performance? Just take a virus. That virus is very infectious and dangerous. We will remove the part from the virus which is harmful for us, but the remaining part as such we will inject or through oral means we will take it inside. But our body immune system will recognize that something came inside which is dangerous. So naturally our immune system will keep producing antibodies against it and we will get immunity. So when the actual infection comes of that particular virus, we are ready with the weapons because we know them, how they are going to infect us. We know them about in advance. So these vaccines will give us better increased or improved immunity against this deadly disease causing bacteria and other viruses. That is the secret of vaccines. When we look at the use of microbes in the field of medicine, it is basically into two major categories: preparation of antibiotics and preparation of vaccines. Let's see them one by one. What are called antibiotics? Antibiotics are product of microbes itself, that the microbes are producing some chemicals or proteins that will kill the other microbes. Examples: streptomycin is produced by a bacteria and penicillin is produced by a fungus, Penicillium notatum. So these things, penicillin, streptomycin, these products will kill the other microbial growth. That means we can use this as a medicine. Am I correct? That is the first use. Next one is preparation of vaccines. What are vaccines? Let's say there is an infectious virus. We will kill them or we will make them inactive by removing the dangerous part, and we will that entire microbe or a part of it, we will inject it or take it inside so that our immunity will be induced. So that when this actual living organism comes, we will have better immunity against these fellows, where you will have better antibodies already produced, so that we can kill this infectious disease causing microbe the moment it gets inside our body. That is the use of microbe. Let me tell you one more time. Let's say this is a virus causing disease, and we will remove the genetic material alone, which is actually the dangerous part, and this alone we will inject inside. So our immunity system will recognize this fellow and we will produce antibodies against it, and we will kill it. So the antibodies will remain in the blood itself. So when the next time the actual virus comes and attack our body, we are ready with the antibodies to kill this virus instantly. So this is a working mechanism behind the vaccines. Let me conclude. In the field of medicine, microbes are helping us in the preparation of antibiotics and vaccines. If you look at the types of vaccines, vaccines are all of different types. Live attenuated vaccine is a type of vaccine where we are using the live organisms, but we remove the dangerous part. Example is MMR, measles, mumps and rubella. Against these three diseases, we are using MMR vaccine, or BCG vaccines. Bacillus galmatae-guerin. In the second type of vaccines, they are killed microbes. That means we take the infectious virus or something like that, kill them and inject it inside our body. Example is poliovirus. The vaccine we actually give is the poliovirus itself, but it is killed one. So that actually will trigger the immune system to fight against the poliovirus. The third part of this type of vaccine is subunit vaccines. Example, hepatitis B vaccine. We will purify the antigenic part of the microorganism alone and remove the organism, take the antigenic part, disease causing part alone, and take it inside by injection or oral method. This will improve the significant amount of immunity against this particular disease causing microbe itself. When the infection comes, we will be successfully able to handle them because we have already made, our immune system already made antibodies against this hepatitis B antigens. The fourth type of vaccine is toxoids. What are toxoids? We have in the last one, subunit vaccines, we are exactly taking the antigenic part, but it is active. This time we are taking inactive antigens. Example, TT. We all know whenever we have a wound because of a rusting iron nail or something like that, we go for taking an injection called TT injection, tetanus toxoid. That actually prevents us from a fever called tetanus. It is a very dangerous one. So by taking an injection of TT, we are actually getting immunity against the tetanus. If we are looking into the types of vaccines, they are into four. The first one is live attenuated. That means that particular microbe is alive, but the disease causing area alone we removed. Like in an antigenic part, let's say this is a disease causing antigenic part, we will remove this alone, and this entire live bacteria or virus we will use it for injection. So our body will develop antibodies against it, so that when this actual infection happens with the antigen, we will immediately destroy this. That is the live attenuated. Live, but they are not dangerous. So next one is killed one. Example is polio virus. So we take a vaccine orally, polio vaccine, that is a killed polio virus itself. They are not altering anything. We will kill it and take it inside, so that our body will develop immune system, will be induced against this polio virus. So even if the actual virus comes, we are ready with the weapons, antibodies to kill this instantly. And the next one is subunit vaccines or purified antigen. This time not the organism, but this antigenic part alone we will extract it and inject it. So antibodies also will be produced, so the immunity remains active against this particular organism that has this particular antigen. That is the technique. Example is vaccine against hepatitis B. The last one is toxoids. That means this antigen itself we will damage it and we will give this to that particular organism. So damaged or very purified, inactivated antigenic part we will inject. Example, toxoid, tetanus toxoids is an example for that. And next point we should discuss is why we should take all these immunization inside our body. Because in this world we are experienced or exposed with so many diseases over the time period. So we have to be prepared. Our immune system should be prepared to attack all these things. Of course, even if they get the disease, we get the immunity, but it will take time. Sometimes we may get some severe damage inside our body. So it is always better be prepared. For that purpose, we have to take the vaccines in a period of time schedule. So that is the program we are discussing next, immunization schedule in India. So there is a system developed by Edward Jenner in early centuries. He made a conclusion that the systematic intake of these vaccines, we call it as immunization schedule. So what we will see, the next one is what is called immunization schedule in India. So let's see what is the immunization schedule we actually follow. Table 22.8 is a book back question till 9 to 12 months. But still, you have to refer table 22.8. In that, you have a bad news also, that the age of 16 next year for you, you are going to get one TT and a TAB injection one more time. So a second booster. So if you look at the schedule, newborns will get BCG, Bacillus calmette-guerin, first dose. 15 days oral polio will be given, first dose. 6th week DPT, diphtheria, pertussis, tetanus plus polio. And 10th week, 14th week we are going to give neither doses, and 9 to 12 for against the measles we are going to give a vaccine for the first dose. And remaining you should go through your textbook and refer it. What is the next thing you should do with this? I will send this notes to you in the form of PDF. Please copy that to your fair notes. Plus study the points to remember. After this end of the lesson you have a key points and glossary, also points to remember. These two also please include in your studies, because if you're looking at the question papers of last year, that it is a practice that few questions are asked from book inside also. So where you have to study at least the points, key points and points to remember. I mean the glossary and points to remember, both these things along with the book back you have to study and prepare for your examinations. See you in the next class, session 3, bad effects of microbes, in the next video. Until then, stay safe and eat well, stay healthy. See you.